Good. Okay, so good morning, everyone. I think it's about our 17th seminar. I'm not 100% sure of numbers now, being a very bad numbers person. So it's going to be very interesting for us to have a scientist and numbers person potentially speaking to us from a completely kind of different perspective, I think, from where we've headed somewhat in the last couple of presentations, where we've been thinking mostly about living with this pandemic, and it looks like this is going to be well, maybe I shouldn't say that. I'm kind of like a slightly doom and gloom kind of prophecy that this might become part of our existence. So it'll be very interesting to get a scientist's perspective on the pandemic, and I am going to, in a minute, hand over to Margaret Lowies, who is an environmental scientist, you know, broadly speaking. She'll introduce herself in more detail. Just our usual process. What we'll do is give the floor over to Margaret. I think floor is a weird metaphor, but give the screen over to Margaret, and she will present a presentation to us, and then in the chat session, as you know, raise questions as you go along if they pop up in your mind, or we'll open up for comments and questions and chat after the presentation. So with that, over to Margaret. Nice to see you here, and yes, could you maybe tell us a bit about yourself, and then tell us what you have to tell, you know, do what you show us from a scientist's perspective.
Okay. Okay, thanks, Andrea. Yeah, there was a little write-up of what I do, etc. So just in short, I used to work for an engineering firm as a scientist there. And I specialized in water quality and waste. And then mid-lockdown, actually, I moved job to the National Department of Environment, Forestry and Fisheries. So very much a scientist by art and by qualification. I hold 3 professional registrations. So I think, that's about as much as I've got 3 dogs, if you want to know something personal. And so at this stage, I'm an assistant director, actually working on the rehabilitation of wetlands. So it was quite a shift from the engineering firm. So I'm always fascinated by numbers and the actual sciences behind something. One often get media reports that are unsubstantiated or are not quoting the full truth. So every time there's a drought or outbreak of sorts, or whichever issue, water pollution, I'm always interested in the actual numbers and reasons behind it. So for this talk, I kept it very simple. I'm not going to bore you with various statistical models and things like that. I've kept it very simple to the basic statistics. And then at the tail end of the presentation, I just briefly touch on biological systems or components, basically in terms of humans as a species, and then the growth or the trends in biological diseases. So that's what I'm going to do today. So with that, I'm going to jump into the presentation. So the first part is the statistical approach. And then basically what I've got on the next slide is, I think the main question we've all got is, if I get COVID, how likely am I to die? So as of 12th of October, the confirmed deaths were 1.08 million and confirmed cases 37.57. So ultimately, with a situation like this, statistics are very problematic, and very serious decisions were made, for example, on hard lockdowns and so forth, without the basis or the fundamentals of good statistics. So we reacted very early in the disease to actually poor statistics, and we based all our forecasts and modeling on that. So I'm just quickly going to go through the 3 types of fatality rates, just to show you how it was calculated, because ultimately I think fatality or death is the thing that scares all of us the most. So then on to slide 4, it's just a graph of the fatality rate for each nation, and I'll get back to that a bit later. Obviously, Italy was hit the hardest, then Iran, Spain, India. There's a very low fatality rate reported. Whether it's accurate is quite questionable. So those were the statistics as at the 12th of October this year. So case fatality rate is fairly simple. It's the numbers of deaths from a disease divided by the number of diagnosed cases of the disease multiplied by 100. Then you get the percentage or the number out of 100 people that are likely to die of the disease. So this is the most common one used, and I think going back to how likely I'm to die, one would think it's fairly simple. You know, you multiply and divide a few numbers and then you've got the answer. But when you look at case fatality rate, crude mortality rate, and infection rate, the answer can actually differ quite a bit. And depending on which one you use, you will also model your responses differently. So the most commonly used fatality rate is case fatality rate. That's what's often reported in the media, but also quite often reported as fact, while it is actually a fluctuating number. So it's not a concrete number, and definitely not a 100% correct number that's mostly reported in the media. So the case fatality rate relies on the number of confirmed cases. And the first problem is that many cases are not confirmed. So the number of confirmed cases strongly relies on the number of tests conducted. And secondly, it relies on the total number of deaths. And some people with COVID are sick and will die soon, but they have not yet been counted as a fatality. So going on to the next slide, so yes, I've mentioned the media report, and then case fatality rate is not the same as the risk of death of an infected person, even though unfortunately journalists often suggest that. But it's important to note that the ratio between the number of confirmed deaths from the disease and the number of confirmed cases are not total cases. So the more cases and the lower the death rate, the lower is your chance of dying statistically. So in short, the more we test, the more confirmed cases we have, and the more likely it is that the fatality rate of a disease would decrease statistically. So basically, as I said, confirmed cases results in a lower ratio of infection or cases to deaths, and the lower the confirmed cases, the higher the ratio. So one of the things that I've mentioned is when we report, COVID is a fairly complex disease, so there's no set time frame of dying. I can give a personal example. My grandmother of 89 got COVID. She survived, but she still has a lot of fatigue. Her heart isn't working properly. So if she dies now, she won't be counted as a COVID death. So there's also that risk, basically, that a person suffers secondary issues or health issues, and that they only die, let's say, within 12 weeks or so, and then that isn't reported as a positive COVID case death anymore. So yeah, I think that basically summarizes slide 7. So what happens is, if we report on data X, for example, we've got the confirmed deaths, but the scenario might actually be a bit worse, because we don't count the deaths that are yet to happen from COVID. So that skews the statistic quite a bit. So an example is the SARS-CoV outbreak in 2003. So it was initially, the fatality rate was 3 to 5%. But then as the disease got contained and all the people died that got infected, the fatality rate rose to about 10%. So that just shows you it's quite a substantial increase in percentage fatality. And that can usually only be done when you tally up all the final numbers after a disease has been contained. So, and ultimately, with this, it also has a negative impact on how we make decisions about an outbreak, because, as I've mentioned, a lot of the numbers are quite variable, and it really depends on how well we record cases and deaths. So it's quite problematic, especially if you take the COVID outbreak. Decisions were made based on early data from China and Europe. And I'll show you a bit later how the fatality rate actually declined once we, got to grips with the disease. So the world and a lot of scientists did modeling and forecasts based on very poor statistical data sets. So yes, that's one thing. The other thing is, as with case fatality rate, usually the low numbers that you get initially when the outbreaks start, when they get published, it's usually an underestimate of the severity of the outbreak. So the more we test and the more deaths occur, the higher the number is likely to rise, especially in the first phases of the disease. And then it often gives the impression that the disease increases in fatality or death rate. But basically, it's because we didn't have good statistics to start off with. So it's not a fixed rate, case fatality rate, and it's at best an estimate of what is actually going on. Especially with COVID, for example, I think there's estimated 40 to 50% of people at this stage that are not showing any symptoms. So you can imagine, if we add 50% to the total case numbers, how much lower the fatality rate would be for COVID. So on slide 10, this is just a linear graph of the number of confirmed cases, and then the grey lines show the mortality rate basically for the plotted countries. So it's just the statistics as it was on the 12th of October this year. Then the next one, so remember I mentioned 3 types of fatality rates. The next one is crude fatality rate. So that's fairly simplistic. It's the number of deaths over the total population, which one immediately I think would think that's a bit problematic. So it's very easy to calculate, and people like to use it because the total population is usually a fairly fixed constant. It basically measures the probability that any individual in the population will die from the disease, not only those who are infected or are confirmed to be infected. So an example is if there were 10 deaths in the population of 1,000, the crude mortality rate would be 10 over 1,000 or 1%, even if there's only 100 people that has been diagnosed with the disease. So if you have 10 over 1,000, you get 1%, and if you've got 10 over 100, you've got a 10% mortality rate. So crude fatality rate is not a recommended measure of the risk of dying in a population, but it is also often used in the media and misreported. So unfortunately, people sometimes confuse case fatality rates with crude death rates. So a common example is the Spanish flu in 1918. So it's estimated by a few authors that the pandemic killed 50 million people, which would have been around 2.7 of the world population. So that was basically a crude fatality rate calculation. So the number of deaths divided by the total population of the countries affected. So 2.7% doesn't seem so bad then, but it's quite a wrong number, as not everybody in the world was infected with Spanish flu. So if you do that, then the mortality rate seems a lot lower for disease than it should be. Okay, and the last measure that one can use is infection fatality rate. So there you need the number of deaths from a disease over the number of cases. So for that, you need two important numbers: the total number of cases and the total number of deaths. But we don't have the number of cases, so it will also give us an inaccurate number. Okay, and then despite what some media reports imply, the case fatality rate is not the same as, or probably even similar to, the infection fatality rate. So I think in conclusion, often in the media, this fatality rate is reported as a fixed constant. But this really changes over time, and a lot of decisions were made on, I think, a 1 to 3% mortality rate for the disease. But as the disease progresses through nations, the numbers fluctuate significantly. So it depends on the number of tests, the healthcare services available, the type of care. So, for example, the first response usually to a severe COVID case was intubation with a ventilator. And then it was found that high oxygen flow actually does the same job and it's less invasive. So, and then there's been various other responses or medical solutions for COVID that could only be developed as we started understanding the disease. So it's quite logical that the fatality rates would decrease as we basically refine our response to the disease. And then, of course, it reflects a specific population's mortality. So as I mentioned, we reacted on data from China and Europe. And the European population tends to be a lot older than those in developing countries. So in India, most parts of Africa, quite a few Asian countries. So we reacted on a very high mortality rate, which doesn't necessarily reflect the mortality rate that would be experienced in other countries. So I think in conclusion, mortality rate is not a fixed constant, and one always needs to, you need to keep yourself informed as to what the actual mortality rate is, because it's quite a fear-invoking statistic, and you think you could be the 3 out of 100 that could die. But this changes, and it will probably, the mortality rate will continue to decrease as we've got a better response, or as we develop a better response to the disease. So on slide 15, I've just put on a graph of case fatality rates by age, and then I did a quick calculation. So if you are in the 40 to 49 years age group, the maximum estimated case fatality is around 3%. So if you take that, the overall estimated case fatality rate is 3%, of which your age group is 0.4%. So the fraction of 0.4 of 3% equals 0.012%, which means if you're on that age range, there would only be one person out of 10,000 that would die in your age group, obviously excluding comorbidity risk. So unless you are in the age range probably of 70 years upwards, statistically your chance of dying is less than dying of various other natural causes or road accidents. So if you think 1 out of 10,000, it's not that scary anymore. Then just the statistics on South Africa. So we've reported a fairly low fatality rate. And it is, I think, partially because we still reflect a developing nation age profile. So people in South Africa simply do not live a lot longer than, let's say, 60, 70 years. Our healthcare system is quite strained, and we've got quite a prevalence of, let's say, HIV and tuberculosis. So in short, people don't get older in South Africa, and then they automatically move to the lower risk range in terms of age. Okay, and then on slide 17, just the mortality rates as they are at the stage for comorbidities. And I think one thing that was very interesting at this, with this disease was that HIV and TB does not at this stage seem to be a comorbidity. So the high occurrence of HIV and TB informed a lot of the decisions made in South Africa. It was expected that people with a compromised immune system would develop into more severe cases, and that doesn't seem to be the case at this stage. So it just shows you basically when a new disease breaks out over the world, it's very country specific, and we reacted on different countries' statistics and anticipated something that didn't happen ultimately. So as I've mentioned, the standard of care plus more testing results in a lower mortality rate. So for the period from 1 January to 20 February, this graph basically shows statistics for China and how the mortality rate or case fatality rate decreased significantly over the range of about a month and a half. And that was purely because the standard of care improved, but also because people started testing more. And you can see it's a substantial decrease from about 23% down to about 1 or 2% case fatality rates. So over a month, as the statistics improved, the response also had to be adjusted very quickly, because if you sit with a disease that has a 25% mortality rate, it's a very scary disease and something that you really need to contain very fast. For example, Ebola's rate, I think, sits at 40 to 50%, and that's why it's taken that seriously as well. And then on slide 19, it just shows the case fatality rate with the ongoing outbreak as at October 12th. And you could see there was an initial low fatality rate, it peaked, and then it started declining as we tested more and as the standard of care improved. So on slide 20, just for interest's sake, I've listed there's a few diseases, SARS-CoV, with the estimated case fatality rate of 10%, MERS-COVID 34%, seasonal flu in the US 0.1 to 0.2, and Ebola 40 to 50%. So that gives you a feeling of where COVID sits at this stage. So it's not true that it's just as fatal as the seasonal flu, it's a bit more serious. But when you compare it to some of the other disease outbreaks we've had, it's very, the fatality rate is very much lower. And then slide 21 just gives an estimate of the number of deaths from influenza pandemics in the recent history. So obviously Spanish flu was one of the deadliest, with the low estimate at about 24.7 million. And then there are higher and lower estimates by various researchers. At this stage, COVID death rate is sitting at the same rate as the Asian flu and Hong Kong flu. So that's basically where COVID measures, but it's very far in terms of numbers from the quantity of deaths that you got for Spanish flu, for example. Okay, so that's the statistics. I think, I kept it fairly simple, but what I ultimately want to emphasize is that your modeling and your forecasts are only as good as your statistics are. And your statistics rely on accurate recording of cases and deaths. And ultimately, I think the question is whether we should have had COVID or let it run a bit longer before we made the harsh decisions that was made for this disease, the hard lockdowns and restrictions. Because from a scientist's point of view, we simply did not understand the disease well enough before we started modeling and forecasting worst case scenarios. And now we've got a much more informed basis to work from. So I think it was 3 or 4 days ago, it has been shared and distorted and selectively quoted by the media, but the World Health Organization released a statement basically saying that they do not recommend hard lockdowns anymore as the first response to COVID. So that's quite interesting. So it seems we're starting to almost moderate our response to COVID as we start to understand it better. Okay, and then just a biological approach. So when we get sick, it's usually a virus or pathogen that makes us sick. And those live in animals, so it's a zoonotic disease, we call it. So just the last few slides, I'm just quickly going to go through some interesting information. So from a biological perspective, from the study that's quoted there on the slide, they've basically plotted the global number of human infectious disease outbreaks and the richness of casual diseases from 1980 to 2010. So what it basically plotted on the upper left-hand graph was the total global outbreaks on the y-axis and the total number of disease-causing outbreaks in each year, which is indicated on the right-hand y-axis. So I think, in short, to summarize it, the number of outbreaks and the disease richness, which refers to the mutation of infectious diseases. So diseases are mutating, and we've got a wider range of pathogens and viruses that can affect us, and the outbreaks are increasing. And then graphs B, C, and D just shows the distribution of human-specific zoonotic species in infecting us. And then C shows fungi, parasites, protozoans, viruses, bacteria, and graph D shows vector-borne and non-vector-borne diseases. So I think, in short, to summarize, the number and richness of the number of outbreaks and the richness or diversity of infectious diseases are steadily increasing over the years. So these authors basically found that the richness of casual diseases still exhibit a significant increase as to the number of outbreaks and richness of casual diseases for each subcategory of the pathogen taxonomy. Then on 25, these are basically just maps which shows the global outbreak diversity for nations over the world over time. So the dark orange and red, you can basically see how it shifts over the various countries as time progresses. And that's basically the orange and the red is an increase in the diversity of infectious diseases. Then usually in terms of emerging infectious diseases, it's dominated by zoonosis or zoonotic origins, of which 71.8% originates from wildlife. And then 54.3% of EID's events are caused by bacteria or Rickettsia species, reflecting a large number of drug-resistant microbes. And then what's interesting is the species richness and the occurrence of these disease-causing pathogens and viruses are strongly clustered around the lower latitudes, which is 30 degrees north and south of the equator. And then the problem is you also find a lot of lower income or developing countries also within those latitudes. So you have people that often rely on wildlife as a form of subsistence. And then countries that usually have a very poor response and data on a disease due to the substandard healthcare systems, etc. Then slide 26. This country had a this publication had a slightly different finding. So they found that the concentration of EID events are actually decreasing. Their data set stretched from 1940 to 2000. But they also found that around 60.3% of EID events are caused by zoonotic, specifically wildlife pathogens. Okay, and then the last map, I think, on slide 29. So map A is zoonotic pathogens from wildlife. So you can see the yellow and red clustering there, specifically also around the lower latitudes. Here's the zoonotic pathogens from non-wildlife. So you can see the comparison between A and B. There's a lot less red and yellow on B, confirming that it's predominantly from wildlife that we get these diseases. C is drug-resistant pathogens and D is vector-borne pathogens. You can see it's following a similar pattern for all of the types of diseases. So just some things to consider. I tend to think of as a species, and We don't act obviously similar to any other species on the planet. So things to consider is we currently live too long. So nature would have probably killed most of us by around 40 years of age, which means that when we do get sick, our health is usually compromised in one way or another. So it's not the strongest that survive. We've got good health care and medicine. So the human population is by default more susceptible to a lot of diseases and pathogens. Mostly because people live longer and can have more underlying diseases for longer without dying. Then a lot of us are not fit. We are in bad shape, and we've seen with COVID the morbidity rate for cardiovascular disease, for example, and diabetes. A lot of these illnesses would have also killed us a lot earlier, and then, as with the first bullet point, we are living with quite a high standard of life despite these diseases. But when something like COVID happens, we've got a large portion of the population that is quite vulnerable to die. Then the third bullet point is we migrate beyond natural range of habitat. So the more we basically invade natural habitats, the more frequently we've got contact with disease-carrying species. And then we tend to spread the disease a lot further. So we don't act like a normal, let's say, a herd of antelope, which are restricted to a certain area geographically and to a certain habitat. We invade habitats, get into contact with various disease-carrying species, and then carry those diseases out and spread them quite widely, quite quickly. So we differ quite a bit in that from how other animal species behave. We do not self-regulate as a species in terms of biological thresholds. So obviously we are reaching the Earth's carrying capacity for various components. We are approaching quite a few thresholds, but we tend to just recreate more resources, which also then leads to a destruction of biodiversity. So we infiltrate and transform habitats, let's say, for agriculture, and we lower the biodiversity. And when biodiversity in a system is decreased, it impacts the organisms and the animals and their health in that ecosystem, which makes them also more susceptible to various diseases. So we are actually causing other species to also be more susceptible, and then we interact with them, and those diseases are carried over to the human population. And then I think lastly, as we've learned from China, we often eat species that's not in our natural diet, specifically zoonotic species or animal species. So those are just some of the things to consider from a biological approach, and questions that we need to consider. And I think these last bullet points, linking back to the graphs, etc., that we are probably going to see more and more outbreaks similar to COVID. And if we don't adjust our biological approach and don't act on proper data, we are probably going to have the wrong response to a disease, or an uninformed response. Yes, and I think that's that basically from my side. Thank you for listening. I know sciences isn't always the most exciting, but yeah, I always find the numbers exciting and the facts.
Margaret, thank you. I have this terrible feeling that we're sitting on a kind of a bomb waiting to go off under our feet from those last few bullet points. But thank you very, very much for a very informative and quite terrifying presentation. I see that I haven't looked at the chat, but I'm going to have a look now and just see whether there are questions, and I will read them to you, and then maybe you can respond to that person, and then we'll see if that person then will come up and respond to you. So Bernadette Snow is asking you, so are you indicating first that we do not make decisions based on the best data? And second, an assumption that media understand the differences between the measures they access. So are you assuming that media actually understand the differences between these measures? Yeah, I think.
Yeah, I think usually scientists act on good data, you know, these multiple peer-reviewed studies. I think it was just the rate pace at which this virus surprised us, which led to bad statistics. And usually when you've got bad statistics, you approach decisions with caution. And I think, yeah, there might have been a bit of an overreaction or some very drastic decisions made on poor data sets. And then as to the media, I think to ultimately also hold them responsible, because they've really catastrophized this disease, and there was the sense of doom across the world. And I think ultimately it's difficult because journalists report to the general public, and you need to keep things fairly simple. And a lot of those numbers were quoted from these statistics. So I guess you can't have in a normal update about a COVID fatality or whatever, have an entire, you know, disclaimer footnote explaining all the statistics and the possible defects in the data set. So I'm not blaming journalism as such. I think perhaps as individuals, we also need to take responsibility to inform ourselves. And then a leader that's also presented on this forum shared quite an interesting comment in that we ultimately create narratives. So I read something in an article and I quote the worst number that I read in the article, or the one guy that died from a neurological response to COVID. And in stories that people tell each other, they like to give you the most, you know, catastrophic version or the most interesting version. And I've encountered that quite a bit when I talk to people, that they selectively quote things or don't understand what is quoted. And then as people talk to each other, we create very catastrophic narratives. So ultimately it does, I think, go back to the individual as well to stay informed and not just rely on media reports.
Yeah, hard though. So if we have a look at the chat, I hope that I'm seeing all of it, but there's a response from Gary saying, surely by the time we understand the disease, it's too late to respond effectively. Thoughts on that?
I think, yeah, I'm not saying let it run for a year, but I think one might need just a longer observation time. So if you remember the slide that I've shown you in terms of fatality rate across a month and a half, the moment we adapted our response and standard of care to the disease, the fatality rate dropped from 23 to about 3%. So the scientist in me, which it doesn't sound very humane, would say let it run a bit and get better data before you react. because decisions are made on information, and if we don't have the information, we make decisions. So perhaps if there was a more cautious approach, let's say starting at a level 1 or 2 lockdown and tightening it up. But I mean, it's so diverse across for us, hard lockdown was to prepare our healthcare system. In other cases, it was purely to flatten the curve of the virus. So, yeah, I think to conclude, the scientist in me said we should have let it run a bit longer. It does go on a logarithmic scale, but I think we would have still been able to flatten the curve. And when you look at the mortality rate, it's still very low compared to a lot of other diseases. So yes, we could have probably afforded to get just a bit more data and a bit more understanding before we reacted like most countries did.
I think perhaps a sort of a slightly counter position to that, and I'm going to jump in here because I'm, audible. In a way, the neurotic in me, or maybe the philosopher neurotic in me, would have gone the other way, exactly the way that we did go, because we didn't know whether this was going to be a 50% or a 0.3% of a kind of, we just didn't know in the beginning what it was going to do. And my thinking is better just lock down rather than see what happens in case it's a 50%er. So if you're in a position where you have no clear or good data and you really do have to act now on the basis of no knowledge, my feeling was that I thought the lockdown was a good idea. So a slightly different perspective, but
yeah. Look, I guess it also, it depends on from which angle you come. I think a lot of us are in academia, I'm in government, and we got our full salaries. We did not need to go through the anguish of when I left Oricon, they were cutting salaries by 50%. So I think a lot of us are sitting in slightly privileged positions. I've personally, lost a friend during COVID to suicide. And a lot of those were directly attributable to the economic situation that she was in and things that weren't available under lockdown. So, and I think the economic impact will only really be quantified properly once this disease has run its course, and probably only in 2-3 years' time will we truly understand that. So I'm not saying let the disease run freely, but when you look at a logarithmic scale, you can still afford, let's say, 2 weeks or 4 weeks period before you clamp down in a hard lockdown just to collect more data. I think I completely understand the counter argument that if you've got no data, rather be safe than sorry. I think perhaps one should find a balanced approach between the two. So understand the disease a bit better, perhaps start with the lower level of lockdown. And then as you understand it better, if you see it's that serious, proceed to hard lockdowns. So yeah, that's basically it. So there's definitely a midway there somewhere.
Okay. Yeah. Gary wants to know about excess mortality. Gary, you say here don't agree. Do you want to actually come up and so we can hear your voice a bit instead of mine for a bit, and tell us what you don't agree with? Hi.
Hi, Andrea. Hi, Margaret. Thanks for the input. I mean, I think, and it goes to the comment that Bernie's made in the thing, is that this is a systemic thing. You're not going to have the data that you need in terms of what you're looking for. You're never going to have it. And we know pandemics fairly well. So you have to, and we know that in pandemics it's critical that you act quickly. And if you don't act quickly, then you increase it by multitudes in terms of the impact on your population. And in South Africa, particularly, the issue is that we don't have an informed populace in terms of how to deal with pandemics. Versus Japan or South Korea, where people automatically started putting masks on when they heard of the disease. In South Africa, we didn't have the same response. The next time round, we would have a different response. But this time around, and it was critical that we locked down areas to prevent the transfer of the disease so that we could manage it, because we don't have a very good healthcare system. So waiting a week, waiting 2 weeks in these situations is critical in terms of how far that disease spreads and then your ability to deal with it later. You know, so that's the one point I wanted to make. The other point is that COVID is the first sort of global response that we've had. So I think it's 185 countries, whereas if you look at MERS and bird flu and that, it was only around about 30 countries that it impacted. So it's the first time that we've had a global disease in modern time where we could actually study it and measure it. And so when we're making comparisons to flu and influenza, it's not apples to apples comparisons that we're making. We have never studied influenza to the same extent that we've studied COVID. So you can't, you know, in terms of testing, the amount of testing that we've done, et cetera, et cetera. So, I mean, I think you using the case fatality rates in terms of what you're measuring, and the case fatality rate, as I understand it, is impacted by 4 factors. The one is the testing rate. The other one is the medical facilities that we have. The other one is obviously the individual response. And then the final one is the recording of deaths. So I didn't see that in your approach earlier. And if you look at, and that's why I've asked the question about excess deaths, is if you look at our normal death rate versus our actual death rate over this period, I think we've lost 18,000 people to COVID, but we've lost 48,000 or something like that to excess death. So above the norm for this time of the year. And so the suggestion would be is that extra 48,000 is actually untested and tested cases of COVID that have resulted in the system. And so that's why, yeah, I mean, I understand your caution, but I don't think that there's a middle ground on this thing. In the case of pandemics, you have to act quickly to prevent the spread of it, and that is the cautionary approach. The impact on the economy and that is understandable, but that's stuff that you can see. We can't actually project that if we didn't act, how many people would have died. And that's the problem in terms of how we view the issue. We're viewing the issue in terms of things that we see, but actually we have to make decisions based on things that we can't see. Yeah, so that's my input.
Okay. Okay, to come back to the second one is I did address in the presentation the recorded deaths and those not recorded, and how they can skew, let's say, case fatality rate. So what I've mentioned basically is the date that you've recorded your fatalities and that you reported against the number of cases that you will have ongoing fatalities not reported. So I think that's one of the statistical issues that's been acknowledged in the presentation. And yes, I think it all comes back to reporting. So what I'm saying is that the statistics are poor, and the understanding of it is poor. And yes, I take your point. I think that it's the first pandemic of the scale that we will get, or that we got, but also I think we will start dealing with these more and more. And if we've got this reaction every second or every third year, and in 50 years' time, we will not have an economy left. So I think the approach going forward is we've learned our lessons. And it's true, as you said, it was the first of this scale that we've had, although the mortality rate is currently sitting at the same numbers as MERS and COVID. So I think one obviously needs to moderate between the economic impact and your healthcare impact and the number of deaths. And I think coming from my side, ultimately it's difficult because we are all people. I've had several COVID cases in my family. But when you approach it from a purely statistical point of view, the numbers are often not significant or statistically insignificant. So it's difficult because you've got a personal response and you're worried about your family. And it is a scary disease for a lot of people, but statistically at this stage, it doesn't compare to the mortality rates of other viruses that we've had over the past. So yes, and then sorry, just back to your first question. Can you just repeat your exact point that you wanted to make?
It was hang on a moment. We're kind of running a little bit short on time, so I would like to know before we continue here whether Bernadette would like to explain a little bit more about what she's saying.
Hi there.
Hi, Benny.
Sorry, I'm sitting outside and to try and get some rays of sunlight for a change.
Awesome.
So my
Lovely
background. Yeah, so I think I'm sort of erring and agreeing with some of the comments that Gary have made. And Margaret, I fully understand the response from a very scientific perspective in terms of data, precautionary approaches, and that there have been some severe intended and unintended consequences based on the decisions that were made. However, I think it would be a little unfair to say that, you know, we could have thought differently or done things differently, because hindsight is always, it's valuable. So my question would be, is don't you think we have learned from this that there wouldn't be the same response if this had to happen again as predicted by the biological factors that you mentioned, would be my question. And yes, as individuals, we need to empower ourselves and look at the correct knowledge, but not all of us have the means to understand, and we put belief in scientific knowledge. And what you're showing now is that technically we shouldn't have belief in scientific knowledge, and there is already a lot of doubt and suspicion around scientific knowledge out there. So, you know, maybe I'm not making sense, but I really think it is a lot more complicated than we should have waited 2 or 3 or 4 weeks in making decisions. Because I think the decisions were made at the worst case scenario, the best intentions for the country and the people. However, what it has shown us that because of our social and our political and our economic context, the consequences have been dire in other ways. And that when we respond to global pandemics, context matters. And we also need to look at what has happened in the rest of Africa with regards to why have we got additional immunity is a possible reason why there could be differences in death rates and so forth and so on. Also looking at that the suspected species or carrier was from Africa, so we might already have an immunity to it. You know, there's a whole lot of things that are being thrown around and a lot of research that needs to be done. And I do feel that saying that we made a knee-jerk reaction based on poor statistics or poor data is a little bit unfair in this system, because it was made with the best intentions, based on the best available data, and taking a precautionary approach, not based on the data, but based on the lives of the citizens of South Africa. So this is just my view. And therefore, you know, sort of going through the whole thing is that it's really a lot more complex, and just stating that if we had better statistics, we would have done something differently, or better biological data, we've done something differently. But what I do hope is we've learned from this situation, I think, in a nutshell.
Okay.
Thanks, Bernie. Margaret, a tiny response, because I see that we are seriously out of time.
Okay. Yeah, Benedict, I think, look, this presentation was very superficial, and it's very, you know, there are a number of different spheres and things to consider. So what I've spoken about today is mostly statistical, and I agree you need to have a humanitarian approach to it as well, and people's lives do matter, even if you lose one or two. In terms of letting the disease run, I think 1 to 2 weeks is just an example at this stage. One would have obviously had to run based on the statistics that you've got, how quickly the infection would have spread, things like that. So I think ultimately, going back to statistics, I think, yeah, we've definitely learned a lesson. And our response will be different the next time. So I think based on this presentation, it just highlighted that statistics were poor, and the only way that you can improve statistics is to get more data. And that's how statistics work. It's debatable on how long you should have a disease run or when you should institute a hard lockdown. It's much more complex, as you say. So I think for the purpose of this presentation, it focused purely on the basic statistics and the basic biological trends that are emerging across the world.
Okay, thank you. I mean, that's something I would like us to end on, and I'm glad that you raised it, because those last bullet points in your final slide, I think, is something that if we go back, if somebody would like a recording of this presentation, I have recorded it successfully this time, and if I go back to it, then that's the slide particularly that I would want to go back to and think about, because sorry, things change.
No, I just ended the yes.
Like, you know, environments just. So yes, I would very much like to think about those implications, those broader implications of the things that we do as humans. And I'm actually saying this to myself because, as you know, I'm flying to Belgium this evening as a human, doing the kinds of things that are actually on your list. So yes, very interesting stuff to think about. I just want to say thank you very much for a massive amount of food for thought. And I also don't want anybody to go away thinking that science is useless or science is junk, or statistics are, as you say, not perfect, but that doesn't mean they're meaningless. We've had this conversation quite a bit in this seminar, particularly at the beginning, where these questions were raised right at the beginning. How can we make decisions on the basis of numbers that are not accurate? They're not accurate, they're not perfect, but that's what we have, and we have to make decisions one way or another, and then we can look back on them and say, okay, well, seeing how things panned out, that decision maybe wasn't the best thing to have done in those circumstances. Had we had the information that we have now, we might have done something differently, but now we'll do something different. So I think you're right. We'll learn from our, hopefully learn from, you know, increased information, increased science. So I still want to put a vote of in praise of science there. I'm very, very afraid of this idea that science will become, one becomes suspicious of everything scientists do, and then in the end, you can say what you like, because you know, you can supposedly make science confirm anything and then nothing is true anymore, and there's no substance to our knowledge system. So I really don't want to go there. But yeah, I don't think we're going to do that. So thank you very, very much. Yes, and thank you everybody for being here, and perhaps our usual cheerio. I'm hoping that I will see you again on Friday, but from a different place. Hi Glenn, hey Gary, hey Sharon, hey Bernadette. It's lovely, I know it's not real, but lovely see behind you. Okay, it was great to see you, and yes, we will see you next week from another place. Bye.